Bismillahirrahmanirrahim. So our today's lecture is about the DNA extraction. How can we extract DNA, like for any type of molecular type of molecular activities. We are in need of extracting the DNA from, for example, if we extract the DNA from the cell of an animal or a bacteria or a plant, so we need a proper procedure for that. So here we will discuss only the basic concept about the extraction of the DNA from any cell. The microeconomics or the basic principle of DNA extraction is the same for all types of the cells, either plants, animals or microbes, but the protocols gets differentiated during, and people use different types of protocols and chemicals for that. So here we will discuss only the basic concept about the DNA extraction, that what we actually do for extracting the DNA. So our DNA extraction topics will include cell lysis, precipitation, purification, and in the end I will add the references from where I made these slides and lecture. The first step in the DNA extraction is the lysis. Then the second step is precipitation and purification. If you have watched the video I have shared in the Google Classroom, you can easily see that in the first step what we do, we have to lyse the cell, break down the cell into pieces, so that we can separate the cell from each other, because for extracting the DNA we need a single cell for that. So in the first step what we do, we break down the cell, we break down the cell, for example we break down the tissue into different parts, and then we extract one cell from that, and from that one cell we will be extracting the DNA. So what we do in the first step, we have to grind or crush the DNA by different mechanical methods. In case of plant cells, we use as you can see in the video, we were using what, a grinder type of thing in the test tube. First we added a liquid nitrogen to freeze the cell, which was making the cell able to break down easily and make the cell in the tissue in pieces, and it was easy for us to extract the DNA from the cell. Now in the second step, what we do, we add some chemical to the break tissue or the broken tissue, so that we further separate the cell. And the chemical, we usually add a saline solution or salt solution. So we add a saline solution to that, so that the cells get separated from each other. More separation is performed in this second step, because in the first step the breakdown of the tissue occurs and the cell are differentiated and the cell are separated. But in the second step what we do, we add a saline solution, so that the cell gets further separated from each other. In the third step and the last step, what we do, we purify the DNA, because the DNA is in soluble form, and the DNA is easily soluble in water solution or saline solution. So then we purify that solution with the help of alcohol. These are the main steps of the DNA extraction. Now continuing to the lysis, which is the first step. In this process, the cell and the nucleus are broken open to release the DNA inside out. What we do, we broken down the DNA, the nucleus and the cell, because in the very first step what you have to do, the cell, as we know, is covered from different sides, from completely by the cell membrane, and the nucleus is also covered by a membrane. So what we do, first we have to break down those membranes and those protections, so to make the DNA out from the cell. There are two methods used for that. The first method is mechanical disruption. Uses, and the second method, mechanical disruption, we usually use grinders and you can say crushers or grinders, any type of mechanical method, so which can break down the cell into pieces. In the second method, we use detergents or you can say another type of ingredient or chemical which is called as a proteinase K enzyme to free the DNA and dissolve cellular proteins. We usually do it in two methods. Lysis is done in two different methods. The first one is the mechanical disruption, and the other second thing is usually the use of detergents or enzymes. पहले मेथड में हम लोग क्या करते हैं, डीएनए का जो ब्रेक डाउन होता है उसमें हम लोग मैकेनिकल डिसरप्शन करते हैं. मैकेनिकल डिसरप्शन में क्या होता है कि हम लोग डीएनए को पीसेस में डिवाइड करते हैं किसी भी मैकेनिकल मेथड के थ्रू. मैकेनिकल मेथड में क्या होता है कि मैकेनिकल मेथड में हम लोग किसी ग्राइंडर को यूज करते हैं. ग्राइंडर जो होता है या जिस तरह हमारे साथ जूसर होते हैं उसमें हम सेल्स को डाल के उसमें जो है उसकी ब्रेक डाउन करते हैं ताकि सेल की ब्रेक डाउन हो जाए और उससे डीएनए बाहर निकल आए. ठीक है. एंड द सेकंड मेथड, उसमें हम क्या करते हैं, वी यूज डिटर्जेंट्स फॉर दैट. डिटर्जेंट्स क्या होते हैं, जो मतलब हम घर में जो डिटर्जेंट्स यूज करते हैं कपड़े धोने के लिए, सर्फ वगैरह, तो हम इसमें जो है जो यूज करते हैं उसको हम लिक्विड सोप कहते हैं. ठीक है. लिक्विड सोप जो भी सोप होता है जो लिक्विड फॉर्म में होता है. ठीक है. तो हम उसको यूज करते हैं आइदर, या हम डिटर्जेंट्स को यूज करते हैं या प्रोटीनेज के एंजाइम को यूज करते हैं ताकि वो क्या करे, वो डीएनए के सेल मेम्ब्रेन को ब्रेक डाउन करके उसमें से द सेल के सेल मेम्ब्रेन को ब्रेक डाउन करके न्यूक्लियस को तोड़ के उससे डीएनए को आसानी से बाहर निकाल ले. ठीक है. In the next step, this is the complete lysis, the complete mechanism for the extraction from bacterial cell. So in the first step, what we do, cell is cultured, because this is the very first step in any type of DNA extraction. You have to culture a cell. In the second step, resuspending or buffer lysozyme is added. Like I have mentioned in the previous slide that we use is proteinase K enzyme. In the third slide, lysis buffer is added to break down the cell, and then after that here we can see proteinase K enzyme and RNase A. These both are added, and then the cell is lysed, the cell is break down and then centrifuged, and cell debris are separated and intracellular components are separated. And then resuspending buffer and sucrose irradiate. These are as I have mentioned that there are different mechanisms for different types of cell, but the basic phenomena of every cell, every DNA extraction from the cell is the same. Now precipitation, when the complete lysis step, the DNA has been free from the nucleus, but it is now mixed with mashed up cells parts. So as I mentioned earlier, that the DNA in the second step is usually mixed up with the you can say the solution which we have added. तो हम उसमें क्या करते हैं कि हम उसको उस precipitation से उसे हम निकालना होता है. So the precipitated DNA from this cell debris is separated. First, sodium ions neutralize the negative charge on the DNA molecules, which make them more stable and less water soluble. So then what we do, we add as I mentioned the saline solution. So in the saline solution, the sodium ion neutralize the negative charges on the DNA. So sodium charges जो होते हैं वो DNA के ऊपर जो नेगेटिव चार्जेस होते हैं उनको न्यूट्रलाइज कर देता है, which makes them more stable and less water soluble. तो वो क्या करता है कि वो डीएनए को इस काबिल बना देता है कि वो स्टेबल हो जाता है और वाटर में थोड़ा सा कम soluble होता है. यूजुअली ये वाटर में soluble होता है. In the next step, this precipitation, next alcohol is added, such as ethanol or isopropyl is added, and the cause the DNA to precipitate out of the aqueous solution from the remaining salt, because it is not soluble in an alcohol. As I mentioned in the start, the DNA is not able to dissolve in the ethanol, so we add ethanol in that solution, and then the DNA is not able to get soluble or dissolved in the you can use only soluble for that. तो ये इस काबिल पे डीएनए नहीं होता कि वो उसमें soluble रहे, तो वो जाहिर हो जाता है. In this in the left side you can see the cloudy water. This is the DNA in the form of in water, and in the second you can see rubbing alcohol. So this is alcohol, and the white thing over here is the DNA in alcohol. So purification, the last step is the purification. Now that DNA has been separated, the aqueous phase, it can be rinsed with alcohol to remove the remaining unwanted material and cell debris. As I mentioned that in the DNA we have cell debris and other cell components as well. So what we have to do, we have to separate that aqueous we have to separate the DNA from we have to separate the components of from the DNA, the cell components. For example, you can see the cell components are different, for example mitochondria, Golgi boards and all that. So we have to separate the DNA from that. At this point, the purified DNA is usually redissolved in water for easy handling and storage. So what we do after that, the DNA is redissolved in water for easy handling टू एंड स्टोरेज. हम क्या करते हैं कि DNA को फिर दोबारा वाटर में डाल देते हैं, दोबारा डिसॉल्व कर देते हैं, ताकि इसको हैंडलिंग में हमारे लिए आसानी हो और स्टोर करने में आसानी हो. स्टोर करने के लिए what we need, we have, if you want to store it for continuous use, you can store it on 4 degree centigrade, और अगर आप लॉन्ग टर्म इसको यूज करना चाहते हैं, so you can use it on 20 degree centigrade. After purification, analyze your DNA with PCR and gel electrophoresis. अगर आपने जो है अपना DNA जो है purify कर लिया है and you want it to analyze, then you can you can use PCR and gel electrophoresis. So PCR will produce multiple copies, and gel electrophoresis we can fragment the DNA and find out different types of fragments which are present in the DNA. So it was all about our lecture. These are the references. If you need any type of help, you can also email me or WhatsApp me, or if you want to study in more detail, you can go on these following websites and check your desired question and answer. Thank you so much, and Allah Hafiz.